So today I'm going to be talking about redesigning a plastic degradation pipeline using proteins and also applying that pipeline over to SARS-CoV-2 protein inhibitors. Okay, so um, what we're going to do first is we're going to look at basically the idea behind this, right? Oops, sorry about that. Okay, so we're going to try and derive Pete's variant. This is for the Pete's design. And this is sort of an extension and a collaboration with Joseph's earlier presentation today. And so we computationally derive Pete's variants using machine learning. And then we test those variants. And then our end goal is to basically break down PET plastics into terephthalic acid and ethylene glycol to make version PET. So the overview of our design pipeline is sort of we wanted to approach this from a generalized point of view. So what we wanted to do was generalize this. So for any protein with a prior sequence of protein sequences, we want to be able to create a model that can generate new, better sequences for that protein based on certain metrics. So the metrics we chose here were relative activity and protein stability, which is measured off of melting temperature. So we build off of Burke et al.'s conditioning by adaptive sampling design. And this algorithm is called CBAS. And basically what it does is it mimics directed evolution, where in directed evolution, you take existing sequences, existing proteins, you put them through a screening process to select the best ones, and then you mutate them, and then you multiply them, and then you put them through that screening process and repeat it over and over again. So that's not very feasible to do right now because we don't have actual labs. So what we're doing is we're doing an in silico method where we're doing this on computers using machine learning. So what we do first is we generate sequences using some kind of generative network, and then we store those. We score those sequences using another network, which basically predicts the stability or predicts the relative activity, and then we take the sequences that have high scores, and then we modify the training set. We add to the training set the good sequences, and then we retrain the generative model. And we do this over and over again until we get a great generative model. So first, let's look at the discriminative model or the oracle. So the purpose for it is to accurately predict the property of a protein sequence. So what we do here is we leverage the large amount of protein data that we have and transfer learning to extract secondary structure information and much more higher level information. So from Biswat et al.'s work for UniRep, what they did was they took over 100 million sequences in the UniProt knowledge base, and they trained a multiplicative LSTM, which is basically just it's a recurrent neural network architecture. What it basically does is it takes a variable length sequence and it converts it into a fixed length vector, which is what we want here. So we take a variable length protein sequence and we convert it into a fixed length vector. And from their work, it has shown that that fixed length vector is very good at many diverse secondary tasks, like predicting secondary structure, stability, and many other tasks. So we use this network to predict both relative activity and, melting point. So what we do here is we train a secondary model on top of our generated vector for the protein sequence. Here, this is a very simple model. So it basically takes in the generated vector, and then and it passes through a very simple neural network to generate a melting temperature. And here we use a data set called Meltome, which is a data set that encompasses around 50,000 sequences with melting points ranging from 20 degrees Celsius up to 90 degrees Celsius. And our model, and this comes from various genomes, not just humans, but also other genomes as well. And our model achieves an absolute error of around 7.74 degrees Celsius. And here you can see the loss curve for it. And we also trained a random forest model for predicting protein protein activity. And here we trained this on our PET sequence data, which is around 750 sequences. and it achieves a median absolute error of 7.89 relative activity, and the data set has relative activity ranging from 0, which is the wild type sequence, to around 10,000, which is more optimized sequences found in literature. So we use this discriminant model in addition to our generative model, and our generative model is we tried various different model architectures. So we trained both a transformer, GPT-2, and a beta TC VAE to generate novel protein sequences. So these models basically taken a list of a very large number of sequences, and then they basically allow you to generate sequences from that. So to ensure that the generated sequences are diverse, so we don't want to train it just on the PETase data that we have, the 750 sequences. We want to include sequences that are similar as well. So we take carboxylic ester hydrolases from EC 3.1.1, and we also take our 750 sequences, and we group them together and train our models based on that. Okay, so now let's move on to the second part of our project, which is instead of looking at things from a sequence perspective, we look at things from a structural perspective. So this is more for proteins that already have a large amount of structural data, like COVID-19 protein spikes. So what we do here is to extract information from the structure. We apply basically, this is from from Gainza et al. We have these are called massive fingerprints, which are molecular surface interaction fingerprints. So what it does is it passes a CNN over. So first, what it does is it takes the protein and it models the protein as a mesh. So each individual point on the mesh, as you can see here in this diagram, is a surface molecule of the protein. And so each of these surface molecules can have various different features like hydrophobicity, the electrostatics, and of course the actual geometric data, the surface geometric data, right? And then so what we do here is we pass this basically a patch over the protein, and we generate a single vector from that patch. And then each of these fingerprint vectors basically encode the information from that patch. So what we can do here is we can take our inhibitor, we can take our COVID-19 protein, and then what we can do is generate a fingerprint vector for the patch where the protein could bond, could be a potential interaction site with other proteins, right? And then we can because now we have these vectors, we can easily search through a very large database to find vectors that are similar to this vector for potential bonding partners. So what we do here is essentially a Google search for proteins. We can take the entire protein data bank, generate fingerprint vectors for them, and then compare these fingerprint vectors to our target protein, in this case, the COVID-19 viral protein. And to process these, to make sure we actually try and find potential inhibitors, we basically take our viral protein and we multiply all the surface features by a -1, except for the hydrophobicity feature. And then we generate a fingerprint vector from that. And so after that, after we get all the fingerprint vectors, it's just a simple comparison to find the Euclidean distance between the vectors and to find the smallest Euclidean distance. And that protein will likely be a good bond partner with our target protein. So to confirm all of this, since we don't have access to the lab, we use molecular dynamic solutions. So to set a baseline for our PETase design pipeline, we dock the wild type PETase with a PET ligand, and we use GROMACS to simulate the complex in a water solvent for around 100 nanoseconds. And we measure the bond distance between the carbonyl carbon of the bond to be cleaved and the beta carbon of Ser 160. And so you can see that in this graph right here. So this is a good baseline for us to compare future design proteins to evaluate their stability and their relative activity. I would like to also thank the iGEM dry lab team and also the iGEM wet lab team for their work on this project. Thank you for viewing my presentation.
So thank you very much, Ryan, for your presentation. So now I'd like to address the judges and ask if you have any questions. Could you please unmute and ask them? Thank you.
I have a couple of questions, so maybe I'll ask if the other judges want to go first.
Go ahead, Andrew.
Okay, great. A quick question about your discriminative model. I guess UniRep is kind of the fingerprinting technique that takes a protein and encodes it into something that's that I guess you can sort of ubiquitously apply into an ML model. It's fixed length. So that's kind of latent space in itself, and it's interesting that you're training a discriminative model on latent space, which is pretty unique. So I wanted to know, well, first of all, your validation loss is not very good. It looks like there's quite a bit of overfitting happening. Did you incorporate just like a dropout? I guess you only have a single layer. Did you incorporate dropout in your neural net at all?
I think this one, this loss function, this loss diagram was the model that didn't have dropout incorporated with it. Because if it had dropout, the training loss would be much higher. But this is a mean squared error also for the loss, and so we would take the square root to find the absolute error. Yeah.
Yeah, that which is fine. Your loss will depend on whatever metric you're evaluating. In this case, it's temperature, so your values are higher, which is okay. I was also wondering why you decided to go with, or I'm not sure if you did this or if this is reported work, but why there was a single layer, or I guess 2 layers rather than, even a 1D convolutional net or having several layers with a ResNet, which I think was mentioned earlier.
Right. So we tried using convolutional nets. I tried dense nets, a ResNet, normal convolutional net. They all did not perform as well as just a simple neural network, because this is operating on a transformed latent space, right? So the features that a convolutional layer extracts doesn't really correspond to anything. Basically UniRep, what it does is it generates three 1,900 dimensional vectors. And so if we pass the convolutional filter over that, it doesn't really encode any spatial information. So the convolution is sort of wasted there.
What if you, sorry, I'll let you finish.
Yeah. So that's basically why we chose this architecture.
Did you consider encoding each of the 3-dimensional vectors individually in its own branch and then combining them at the end into your linear layer before you do your dense layers?
Do you mean training, so inputting each of the 3 vectors into its own sort of branch and then combining the predictions
into its own convolution, so you don't, assuming that the 3 1-dimensional vectors are all kind of their own unit. I guess I'm not sure how it works.
No, I've not tried that. That's an interesting approach, but I assume it wouldn't work any differently than using a normal dense architecture, because in literature and in past in a lot of papers that pull off of UniRep, so there's another paper called task assessing protein embeddings. They also use very simple top models. So some of the stuff they use are SVMs, just very shallow models for the very top part. Yeah.
Okay, cool. And then your generative model, I guess you're just sampling your latent space and then sort of blowing it back up into a protein, I guess, right? Or you're starting with a protein and then you're compressing all of them into latent space, and then you're just checking to see with, I guess, a random forest algorithm. I can't remember if you said
for the generative model, or for the random forest was for the discriminative model.
Right, so the generative model, I guess. Are you probing a specific area of latent space based on sort of trends in the latent space for like your target? You compress everything and then you get your map of your latent space. You have good stuff and bad stuff. Are you only probing good stuff, or are you just sort of probing the whole latent space?
So for the generative model, just by itself, it doesn't actually do any of that. But combined with CBAS, like the conditioning by adaptive sampling design, what it does is it basically like modifies our training set. So it implicitly shifts the underlying prior distribution of the generative model, right?
Yeah.
Because we have like newer, better, in quotation mark, proteins. And then that shifts it towards like better, hopefully better design and better performance. Yeah.
I see. That's awesome. Thanks.